And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
All right, it looks like we're live. So thanks, guys, for coming on and being willing to have a conversation. This will be talking about what is it? Universal common descent versus independent ancestry of lineages. So standing for truth, always with the haplogroups and the lineages line. And Aaron,
Aaron, not Aaron.
Aaron is Aaron Ra. Keep confusing that because I had to learn that.
Aaron Ra, is that you?
Yeah, yes, this is Aaron Ra. Yeah.
Hey, nobody warned me about this.
So, would you mind telling us a little bit about yourselves before we get started? Aaron, go ahead.
All right, I'll go.
Do it again. Damn it. Aaron.
Let's see. So, yeah, my name's Aaron. I'm from Texas. I have a background in biology. I'll call it, a bachelor's degree, if you want to call that a background. I'm not like a scientist or anything. I worked for a little while in infectious disease, laboratory science, but went on to different things after that. So definitely not an expert by any means or any stretch of the imagination. I used to identify as a Christian kind of growing up, but I was kind of a type of person who never really actually believed it really, just always full of doubt and skepticism. But as far as my position goes, like, I don't have to be an atheist. Like, I don't like desire to be an atheist. If the claims that like Standing's gonna make are true and make sense, I'm fine accepting those. Like, I have no real stake in this. It doesn't matter to me. I could really go either way. I'm just not persuaded for the theology side of the argument. So anyways, a little bit about me. Go ahead, Standing.
Awesome. Thanks so much for doing this discussion, Aaron, and thanks so much, Tom, for setting it up. I'm looking forward to it. It should be fun. I'm a Christian. I run a channel, Standing for Truth, where we do discussions, debates, videos, mainly on the creation versus evolution controversy. Tom and I have had a couple back and forth discussions, which I both enjoyed very much. So, like I said, thanks for inviting me on, Tom. I really appreciate it. My position on universal common ancestry, I'm certainly open-minded, and I am interested in looking at, I would say, the very best evidence that universal common ancestry would have to offer. And I believe that the very best lines of evidence come in the realm of genetics, because I believe it's in our DNA where the creation versus evolution debate or controversy or whatever you want to call it is actually won. I mean, many people, they don't even realize that the term genetics wasn't even around during the time of Darwin, right? Genes, traits, genetics, that's what's inherited, sperm and egg. Sperm and egg, for example, as you know, they don't pass on a fossil or a bone. They don't pass on geography or a rock. They pass on genetics and traits. So if we want to find out, say, the history of humanity, when it comes to limited ancestry versus universal common ancestry, or whether or not Adam and Eve existed, whether or not there's scientific evidence for an Adam and Eve, I think it's in the realm of genetics, where we have to look to see which version or which position is true. So I think going into the discussion, I would probably just ask Aaron to be open-minded and for him to answer the question, if Adam and Eve were true, hypothetically, what should we expect? Actually, I should say, or ask, what would you expect, Aaron?
I would expect for us not to see any traces of ancestry with us and the great apes, for example, and more closely like chimpanzees, and not to see a branching pattern in phylogeny for all other life on Earth. I would expect to see just straight lines of independent ancestry when we look at our genome, just going back to each separate, I guess you would call it created kinds or something. So I wouldn't expect to see any trace of like the history of certain types of genetic events within the genome, types of structural variants or variants in nucleotide sequences that all paint the same picture that we see from other multiple areas in different disciplines. Kind of, it's a cumulative case for me that kind of adds up, that shows common ancestry. Like we don't have to build a time machine to go back in time to actually witness it. So it's more of a cumulative thing for me, and I don't think we would see what we see if the story was literally true of just kind of two people kind of just kind of supernaturally poofed into existence at a certain period of time.
Right. So I appreciate your response there. And I do agree with what the things you were saying when it comes to phylogeny, when it comes to the relative hierarchies that we see, right? We can look at morphology, we can look at anatomy, physiology, we can look on a DNA level, on a molecular level, we can see nested hierarchies there, whether it's in the mitochondrial DNA or in the nuclear DNA. I would simply respond to those lines of evidence as relative hierarchies, nested hierarchies, I guess, in general, or more so characteristics of design. But I do agree that similarities, hierarchies, phylogeny, they are, those lines of evidence are more so agnostic to the debate, because I think it comes down to lines of evidence that can differentiate between the models, limited ancestry versus common ancestry. So I think the question is, if Adam and Eve were true, what should we expect, right, Aaron? So, I mean, we would expect one female ancestor of all people on the planet. I think you'd agree with that. And then we would expect evidence for one male ancestor of all people on the planet. But I want you to comment on the genetic data that I believe confirms those expectations. For example, in Genesis, a direct prediction or retrodiction would be one male and one female. Now, this doesn't have to be true if evolution is true, but for our model, I will admit that has to be true. And I believe that the genetic evidence in our Y chromosome and in our mitochondrial DNA point us directly back to one female ancestor and one male ancestor. And not to go on for too long here, I would just point to the Y chromosome, Aaron, and then have you comment on that. We know that the Y chromosome is passed on unbroken from father to son. And then every once in a while, as you know, a mistake happens, right? Every time this happens, though, a new branch in the family tree is formed. As you were explaining earlier, these branches that we can see. But if we look at all the branches in the world, Aaron, they go back to a single person, right? Not a chimpanzee, but to a man who lived just a few thousand years ago. That would be, even the evolutionists would refer to that as Y chromosomal Adam. And that's because every male Y chromosome on the planet is nearly identical. And then the chimpanzee Y chromosome to the human Y chromosome is only 70% similar. So how would you explain that genetic data? And secondly, why wouldn't that be, say, convincing evidence for you that maybe there is evidence for a literal Adam and Eve? Go ahead, take as much time as you need.
Okay, thanks. It's not convincing evidence for me, because that's the same thing we would expect under evolutionary theory anyways. It just logically makes sense that we would have to end up going back to an aboriginal source for the Y chromosome and an aboriginal source for the mitochondrial DNA, just based on population genetics, based on ancestry. You've got two parents, they each had two parents. It's going to exponentially grows, and if you go back just 40 generations, that's something like, I don't even know how many billions of ancestors, which obviously didn't exist at one time. So you have to have the kind of this tree going back and then kind of closing back in on itself, to where you ultimately end up at some sort of point with Aboriginal mitochondrial DNA and Aboriginal Y. For what I've, like I said, no expert in this. I sell cars for a living, so I don't have much time to really research any of this stuff. And a lot of this stuff is just gonna be stuff I just kind of remember from reading throughout, out the years and stuff. So I believe the mitochondrial evidence shows that the mitochondrial Eve dates back to about, I think around 120,000 years or so. And as far as the Y chromosome atom, I believe it's around 70,000 years. So I just don't see any issues there. They both models, a creationist model versus a evolutionary model, we would expect the same thing. As far as the differences in the Y chromosome of the human and the chimp, you said it's what, like 70%
Right.
similar?
They discovered that the thing is, if you know, the Y chromosome doesn't experience recombination because it doesn't have a counterpart to exchange its genetic information with. So when they initially sequenced the Y chromosome on the chimpanzee, they figured that the Y chromosome should be the most stable of chromosomes because of this lack of recombination. But they were shocked to find out that the chimp Y chromosome is 70% dissimilar to the human Y chromosome, which, as you know, all human Y chromosomes are incredibly similar. I do want to say one thing, and then obviously you can have plenty of time to comment on that. When it comes to Y chromosome Adam and mitochondrial Eve being a part of, say, a population of whether or not it's 2,000 or 10,000, as far as I know, there's no actual evidence for that. That's just hypothetical, theoretical, because all human beings on the planet are incredibly similar, telling us that we have low genetic diversity. You might be familiar with the out of Africa scenario, where there was an extended near extinction event, which reduced homogeneity. And out of that, say, population of 2,000 or 10,000, I'm not sure, whatever you hold to, you can answer that, Y chromosome Adam and mitochondrial Eve came about. But is there really any evidence that there was a population? And lastly, if you do hold to that view, based on what we know about inbreeding and prolonged bottlenecks, how would a scenario such as that even be remotely feasible, right? Because that would involve severe genetic damage, right? So these Homo sapiens would be extremely genetically compromised, and then we're to believe that they suddenly spread out over the entire planet, seizing dominion over the earth, whether or not they assimilated with, you know, the Neanderthals, the Denisovans, the hobbits, for example, Heidelbergensis. But, you know, that's how the story goes. But do you believe that's feasible? And then why do you believe that's feasible? But I guess before you get to those questions, yeah, how do you answer the Y chromosome dissimilarity between humans and chimps? Take as much time as you need, Aaron.
All right, thanks. Apologize in advance. I'm severe ADD, and about this time of night, my meds wear off. So give me a second to gather the shotgun blast you gave me there. So as far as the differences in the Y chromosome, it's interesting that the Y chromosome in humans and gorillas are actually similar. So there's more similarity between humans and gorillas in the Y chromosome than there is between chimpanzees and gorillas. And from what I can recall reading is just the Y chromosome has underwent over time lots of deletions and then some adding, some transpositions from other chromosomes back onto the Y chromosome. Right. And there is some crossing over that does occur. Recombination does occur like on the tips, but it's very minimal. So there is actually some recombination that occurs. So the fact that our Y chromosome is more similar to a gorilla and the chimpanzees is actually the one that's different. I don't think presents much of a problem there. There's lots to learn about the Y chromosome still.
Actually, real quick before you continue, just so I can clarify, have they sequenced the Y chromosome of the gorilla?
Yeah, actually earlier today I went on to the global database Ensemble, so I could actually pull up the different chromosomes of different types of species, and yeah, you can actually do look at the synteny between the two, or whichever one you want to look at, and so you can do a direct comparison between these chromosomes of different types of species. So it's pretty cool.
Like, okay, so synteny would be like similarity in the gene order of the chromosome. So what was it, similarity in the gene order, or was it like base pair to base pair on the Y chromosome? Like, was it the same type of sequencing that they've done with the chimpanzee Y chromosome that they've done with the gorilla to human?
Yeah, right. And so it's the amount of information that's on the Y chromosome.
Right.
there's more like degeneration in the Y for humans and gorillas versus the chimp for some reason.
How similar was the gorilla to the human Y chromosome?
I don't know. I didn't focus in on that one very much. I just remember kind of coming across it. You could always pull it up.
Like,
Look at it and see.
was it in and around 70% like the chimp and human? Like, was it maybe 75%? Or because as we know, every single male human Y chromosome is 99% similar, even more, right? 99.9%. That's why we know we can trace the Y chromosome back to one male Y chromosomal ancestor. That combined with the mitochondrial DNA and how similar worldwide mitochondrial DNA is compared with in contrast, in correlation with the low genetic diversity, that's how we can conclude, okay, we came from just two people, right? Because that's why the evolutionists had to invent the hypothetical population bottleneck of, say, 10,000. I mean, do you hold to more of a 2,000 population bottleneck, or was it 10? Like, I've heard different theoretical numbers.
I don't know. I have to kind of go by what the consensus is. I've heard Francis Collins, or I read that he wrote, he thought it was about 10,000 based on the genetic data he's looked at and gathered. So him being the head of the Human Genome Project, director of the National Institutes of Health, world leading geneticist and physician, I would tend to trust his opinion on the matters since I'm not an expert in it. So I believe he said around 10,000. And just from other information I gather, I don't really see a problem with the low level of diversity in the human population, because we see the same thing on island populations. There's no difference. So those would go through bottlenecks, of course, speciation events on islands, and they show the same variation as we do species here on the mainland. There was a paper published pretty recently that came out. You're probably familiar with it, that showed all current, about 90% of current species kind of came into being, if you want to call it that, around 200,000 years ago.
Correct.
So based on the ones that they had studied, and they all show very similar, the same percentage of variation within groups.
I guess, and yes, I do want to probably go possibly in depth into that paper, which is good. But before we go on, I guess, that rabbit trail, when it comes to the low genetic diversity, the population bottleneck, Y chromosome atom, mitochondrially, for example, as you know, if evolution and deep time is true, over deep time, millions and millions of years, I should say, any large population like that would most certainly accumulate massive numbers of mutations. And then that would result in, of course, basic genetics would tell us enormous amounts of genetic diversity. So as I was saying earlier, that's a pretty serious problem for evolutionary theory, because what we now know about mankind is that we have, as a human species, very limited genetic variation, right? So then the evolutionists came up with the evolutionary out of Africa bottleneck theory, right? So for us, obviously, if we came from just two people, that means limited diversity is easy to explain. It's exactly what we would expect. But when it comes to the evolutionary model, that near extinction event, I would agree, yes, it's going to reduce homogeneity. So that is a feasible way to explain the low genetic diversity. But the problem is, Aaron, is that it would cause permanent and severe genetic damage, right? That just means, because these enormous numbers of deleterious mutations that are accumulating due to the inbreeding, they would then go to fixation. So my question is pretty simple. I'll say it like this, or I'll ask it like this. How could such a tiny, nearly extinct, of course, they'd be genetically compromised due to the inbreeding, how could that population of, say, 10,000, like you said, we can hold to Francis Collins' view on that, how could they suddenly explode in all parts of the planet and seize dominion over the entire world, being so genetically compromised? Aaron, take your time.
I think you kind of got the order of things kind of backwards. Like we didn't come up with the out of Africa theory or like a bottleneck from the Toba eruption or something because to explain the genetic diversity among humans. I mean, that stuff was already established years ago through paleontology, fossil record and tracing routes out of Africa that way, and also tracing the history of languages, language development. And you can make phylogenetic, if you are, similar trees just based on language that align with the trees made from the fossil record, which later come to align with the trees that we can make from the genetic record. So the chances of all those lineages lining up perfectly seems like pretty good evidence to me that our current understanding is correct. And you asked about.
Well, real quick before, because I want to clarify. Are you saying that they came up with the out of Africa, the hypothetical population bottleneck after they discovered that as a human population we have limited genetic diversity? Is that your position?
No, we've known about that before we were able to sequence the genome. So the genetic data came later, and the genetic data just confirms what we already hypothesized and already knew from the evidence.
Well, from my understanding, because I believe it was Dr. Cy Gart, and I'd have to just double check, the evidence for the population bottleneck comes from genetics, because mankind in general has such significant low genetic diversity, right? In mitochondrial DNA as well, and Y chromosome. So therefore, to explain such limited homogeneity, they concocted the story of the out of Africa population bottleneck, because there's actually multiple theories, right? There's the multi-regional theory, but there's the out of Africa theory. So
it's not concocted. Like, we look at evidence and we see what that evidence seems to tell us.
Right.
And then we try and kind of deduce, hypothetical deductive models to kind of deduce what could have caused the, things we're seeing. And then if we find, and we can make predictions, like maybe there was some sort of catastrophic event that occurred around this same time period that matches the rates of mutations that we see that cause a bottleneck. And then if we find something like that, like this volcanic eruption, then that will help support our hypothesis even more. So it's kind of like with the chromosome number 2, we predicted that in advance, and then now it's not disputed at all anymore that chromosome number 2 was the result of the head-to-head fusion of two chromosomes that remain separate in the great apes.
Apologize, just a clarification. What did you say Cy Gart was talking about?
Cy Gart it was during my debate with Erica. We were going over this same problem, and I asked Erica, how they came up with this population bottleneck, and I believe it was Cy Gart in the comment section who was stating that it's in our genetics where they come up with the population bottleneck of 10,000, some say 2,000, because that's the only feasible way they're going to be able to explain the limited homogeneity that we have as a human species. Because as I explained earlier, if evolution is true, well, we should have enormous amounts of genetic diversity, because these large populations would be accumulating enormous numbers of mutations. And even if I conceded that maybe there was some type of near extinction event based on this volcanic eruption, I do want to go on record saying that there are a lot of scientists and experts in the field that agree that there are some inconsistencies with that volcanic eruption leading to the near extinction event. I would just have to probably provide those sources after. But even if I conceded that, I'm still looking for an answer as to how that hypothetical population could have exploded in all parts of the world, because based on the inbreeding, based on the extended bottleneck, the extended nature of it, it wasn't just one generation or 2 generations or even 10 generations. How did they seize dominion over the entire planet? They would have been so genetically compromised that this just is not feasible, other than storytelling.
So I'd ask, why would they be genetically compromised? What do you mean by that?
So the reason why they would be genetically compromised is because that population bottleneck would include enormous numbers of deleterious mutations going to fixation, because, as you know, inbreeding exposes the hidden reservoir of genetic mistakes. So if there's an extended near extinction event for, say, hundreds of generations or just many generations in general, the resulting inbreeding would cause permanent and severe genetic damage. And yet we're supposed to believe that 70,000 years ago, this genetically compromised population of Homo sapiens suddenly exploded into all parts of the world, thus seizing dominion over the world. I mean, it's not feasible. I mean, we would need some type of explanation for how that could be possible.
Well, I don't understand. I don't understand where you're getting this information from. Like, why would the genome just build up one by one different like single nucleotide changes or whatever the mutation may be, and then just continuously get worse and worse until those things get fixed? I'm not really following your logic there.
Are you disagreeing with the fact that evolutionary bottleneck hypothesis does involve an extended and lengthy near extinction event, and thus resulting in severe inbreeding and genetic damage? I mean, that's just the nature of bottlenecks and inbreeding. Look at the cheetahs today. I just looked it up the other day, and the cheetahs are now down to like 7,000. Because they are inbreeding, they have experienced a genetic bottleneck as well. Their sperm is degenerate. Many recessive mutations are coming to the forefront. I mean, conservationists are looking at them and they're seriously concerned that this population of cheetahs are going to go extinct. I mean, it's a very serious problem. But I don't see these cheetahs being in such a compromised state, suddenly exploding into all parts of the world, seizing dominion over the world. But that's what you're trying to say happened 70,000 years ago with this hypothetical population of 10,000 Homo sapiens. So if we see today that this leads to severe genetic damage, how is this remotely feasible other than, ad hoc, post hoc storytelling, of course?
I think it requires storytelling. I mean, it requires a little more understanding of like molecular biology and genetics, kind of how that stuff works. Like cheetahs, I think we can kind of set them aside because different animal, different types of environment, different types of environmental pressures than humans would have. So a separate topic, interesting topic, but a separate one. As far as the buildup of deleterious mutations, you know what cryptic variation is?
I believe so. Can you explain it?
So, and this is experimentally determined. So within genomes, of course, mutations occur.
Right.
And say for example, you have a polymerase is coming along and it's reading a sequence of DNA, and it's going to produce a messenger RNA, and then it has to stop at a stop codon,
Right.
to produce a certain protein, right? Well, there's lots of instances where you have a read through, and it just keeps on going, and past that stop codon, there's lots of cryptic variation that is not exposed to natural selection. It's just mutations that accumulate over time. And lots of different variation, lots of traits are expressed from multiple genes as well. So you can have lots of cryptic variation build up, and it's kind of stored as like a genetic capacitance, kind of like electric capacitor stores energy, just waiting for the floodgates to open, and
Right.
then be exposed all at once to selective forces, in which can wipe out entire stretches of structural variants that would be lethal, for example. So there's lots of ways for you not to get just some like linear buildup. It's all bad, and then it's going to end up in some homogenized state where the species goes extinct.
Well, I understand what you're saying there. I think that more so has to do with long-term genetic degeneration, like, for example, genetic entropy, which we can get into, right? We accumulate 100 new mutations per person per generation. I understand that, we could argue all day as to how many of those are completely neutral, how many of those are beneficial. But when it comes to the population bottleneck story, we're just simply talking about the result of inbreeding. right? Because I always say that inbreeding, it's like a sneak preview of where we are going genetically as a species. I mean, that's why the reduced life expectancy of, say, inbred children, well, that's reflecting the overall aging of the genome, because what's happening is the hidden reservoir of genetic mistakes, they're being revealed, the ones that have been accumulating over time. So this small population of, say, 10,000, some believe 2,000, and I definitely don't want to beat a dead horse. We can probably agree to disagree and maybe move on to human chromosome 2 after this, but I've never personally received a good answer as to how a genetically compromised population such as that, which I believe correlates perfectly with the highly degenerate population of cheetahs we see today, could have actually exploded into all the world and then seized dominion over the planet, right? I mean, some say they assimilated with the Neanderthals and Denisovans and the other so-called human species. Others say that they killed them off, right? Like I said, there's different theories, multi-regional, out of Africa. So I just see stories coming from the evolutionists, and I think we might just have to agree to disagree on that. I did hear you say chromosome 2 fusion. We can talk about that, but if you want to make a couple comments on what I just said, go ahead. Take your time, man.
Yeah, so I don't think like 10,000, a population of 10,000 individuals would be a super dramatic decrease in potential variation to be, you know, exploited by natural selection as we kind of move to different areas. Like I said, you got these different structural variants that are hidden, cryptic variation, and as we know, it takes very little genetic diversity produces huge ranges of phenotypic diversity. And the phenotype is not relegated to just the actual morphology of the animal. The phenotype also includes on the molecular level, like proteins, for example. Lots of
Right.
molecular processes are going on behind the scenes that you're not aware of. We can test for, and we can see that natural selection is happening within our own bodies, within our cells, and it's selecting for various types of proteins, different structural groups attached to these certain proteins that could, in the right situation, produce a huge benefit. And there's actually really a good paper written on prions from that they were looking into. I wish I had the paper. I can link it later, but it's on yeast, right? And it shows that through a process called congruence, that these mutations, which you say are all deleterious, like nearly neutral, they're actually pre-adapted. So they're not on the negative side of zero, of neutral. They're pre-adapted to be more positive, more beneficial, based on congruence. I can get you more information on that. So if you have like a pre-adapted variants that build up as cryptic variation within the genome, and then you have environmental changes that occur. There's another process called assimilation, which is induced. We've done it on fruit flies, where you expose them to heat shock, and it pulls out these cryptic variants. And then you don't have to use the heat shock anymore. They just keep reproducing this new mutant variant based on this principle of assimilation. I mean, there's lots of good data from molecular biology on how you wouldn't have some buildup of negative, mutations. It's much more complex, much more dynamic than I think you're aware of.
Right. So I'll just have to say, I don't think you sufficiently answered the question regarding the hypothetical, population bottleneck of 10,000. I mean, there's just no feasible way that such a genetically compromised population due to the inbreeding. Because I think you're more so addressing long-term degeneration, right? And we can shift to that, because one thing you did say that I did take note of is that selection happens and on the phenotype, right? Because selection, would you agree selection happens on the level of the whole organism, right? Like, does selection act on phenotype or genotype?
It acts on phenotype, but you need to adjust what your definition of phenotype is. It's not just the morphology of the creature that we're describing, or of the life form that we're describing. It's also deeper than that. So on the molecular level, it's the proteins. The proteins are also that
Right.
get expressed
I
are
agree.
also part of that phenotype.
But one single point mutation on, say, a single nucleotide, if it's not harmful enough for selection to see, then selection is not going to act on it, is my point, right? It has to be phenotype ultimately that selection acts upon. That's why my position is that selection acts on the best beneficial mutations, right? We can talk all day about beneficial mutations that say add new novel information to the genome, for example, or we can talk about the worst detrimental mutations, because then natural selection, of course, they can rid the population of the most deleterious of mutations. But my position is that selection cannot stop the loss of information that's actually immeasurably small due to the low impact mutation accumulation, because as you said, that's actually happening on the molecular level, right? So even if, let's say, even if only 10 of those 100 new mutations per person per generation were deleterious and the other 90 were completely neutral, that alone is still too much for long-term genetic advancement. I mean, there's got to be a type of selection that can select away such low impact mutation. And I understand there's redundancy in the genome, and I understand the things that you're saying. There are mechanisms, right? Synergistic epistasis, the cryptic variants that you're talking about. But ultimately, there's still a percentage of those 100 new mutations that are deleterious. So what type of selection is going to rid the population of those ones, I guess, would be my question, Aaron.
Just to clarify, so Standing, your objection is that if there was a population bottleneck of 10,000, then that would create too much similarity in the genome to be able to repopulate the Earth. Is that your argument?
Right. It would be like the cheetahs that we see today. They're so genetically compromised due to the inbreeding. They're so genetically similar. There's too many recessive mutations that have come to the forefront, and that's exactly what would have happened in the population bottleneck. I believe they say it's about 70,000 years ago. There'd be prolonged inbreeding, and so in that specific case, it's the inbreeding that's the problem, and we can see a reflection of that in the cheetahs today. I mean, they were at 10,000 at one point. I just looked it up the other day. They're now down to 7,000. You know, like conservationists are extremely worried about the
What was the
cheetahs.
lowest? What was the lowest the population of cheetahs ever got?
Right now I believe is the lowest. It's at
No, I mean
it's at 7,000.
At the bottleneck that accounts for the homogeneity they have today. Like, what was the number of individuals?
Right. Good question. Good question. I believe that it's similar to the population bottleneck of humans, right? There's some that say 2,000, 10,000. I've heard some papers that describe them as 30,000, because all they're doing is they're looking at the genetic data. They're looking at the limited genetic homogeneity, and they are
what's the
coming
cheetah's
up
genome
with,
size compared to the human genome size?
so we have 3 billion letters. Obviously, mom passes on 3 billion, dad passes on 3 billion, so 6 billion letters. Are you asking how many DNA letters are in the cheetah?
How many genes that are expressed in cheetahs versus, I think humans have around 21 to 25,000 genes.
Right.
What's the number in cheetahs?
Right, non-coding versus protein coding. I'm not sure how many are in cheetahs. Do you know?
I don't know, but I think that would be.
I got a question for you. So, as far as I know, cheetahs don't intermingle much. They have very close-knit packs, and which is why their genetic deterioration is so much greater, even though they still have a population which would potentially be able to support genetic growth.
Yeah, it's like inbreeding within inbreeding. So
right. So the problem is that they're like in little subgroups, and they're not really diversified in the 7,000 that do exist, which is the problem.
Well, and that's an interesting point too, Tom, because a lot of papers that I have read too is that there would have been subgroups very similar to the cheetah subgroups that we see today in that hypothetical population of 10,000 Homo sapiens 70,000 years ago. It's a very, very similar population bottleneck to what we see in the cheetahs. And I'm not saying that they would have went extinct right away, but we're not going to be seeing these 7,000 cheetahs that are incredibly genetically compromised suddenly seize dominion over the entire planet, like supposedly the 10,000 Homo sapiens 70,000 years ago did. I mean, we can agree to disagree on that point and maybe move on, because I like to touch on everything that's talked about. You did talk about chromosome 2 fusion. I find that a very interesting topic as well. But that's just my position on the population bottleneck. I believe that any explanation or every explanation I've heard is nothing more than, post hoc, ad hoc storytelling. And I understand you disagree with that. But the data is in support of a small population. For example, Adam and Eve, that's what the Y chromosome data tells us. That's what the mitochondrial DNA tells us. And that's just what our genetic diversity tells us in general. And the evolution is my position.
The Y chromosome and the mitochondrial DNA don't tell us anything about a small population. And
well, yeah, because the mitochondrial DNA is incredibly similar worldwide. That's why we can take the mitochondrial DNA and look at observed mutation rates, right? Pedigree-based studies. And that takes us back to a mitochondrial Eve just about 6,000 years ago. And that also goes with the Y chromosome.
Where do you get the 6,000 years from mitochondrial sequencing? Where do you get that from?
Actually, very good question. So we just came out with a video today. There's a gentleman named Mad who I work with on my channel. So we provided a video on mitochondrial Eve with all the papers. So there was an original paper, I believe it was 1996 or 1997, that based on pedigree-based studies, observed mutation rates. They didn't consider like evolutionary assumptions in it, right? Divergence calculations. They just took observed mutation rates. And the calculations that they got back was 6,500 years ago. But there have been adjustments now, because obviously that doesn't fit with the evolutionary story.
Hold on a sec. Sorry. So that was the coalescent time?
Right. But
6,000 years?
Yes, correct. 6,500 years.
Okay. So you do realize that there's a different coalescent time for mitochondrial DNA versus nuclear DNA, right?
Right. Very good. Well, I should ask
if you have a coalescence of 6,000 years for mitochondrial DNA, since it's just a single DNA, single genome, versus the crossing over that occurs between diploid organisms, it would be 4 times shorter than the nuclear genome. So if you're going to say that there was a 6,000 year coalescence for mitochondrial genome, that means the human species is at least 24,000 years old, which would discredit your position.
Right. Very, very good point. So would you agree that the coalescence time, at least before I go into the nuclear DNA explanation, you do agree based on these papers that there is a 6,500 year coalescence time for mitochondrial DNA based on observed mutation rates in the mitochondria? Would you agree with that part?
No, that's not in any of the established literature published in any reputable, relevant journals. It's in the creationist literature.
No. I just have to correct you on that. No, like the papers I'm referring to are in secular journals. I'll post them in, because I don't want to just play our 15-minute video. We got all the sources there. When it comes to the nuclear DNA, I totally agree with you in the fact that the nuclear DNA would take us much further back than 6,500 years ago, which I'm saying the mitochondrial DNA takes us back to. But the question is, and I'll just, I guess, see how well you understand our position. The big question is, Aaron, is where does genetic diversity come from? Now, I know you guys explain the vast majority, if not all, the nuclear DNA differences as a result of mutation. Do you know what we explain the nuclear DNA differences, the millions and millions of nuclear DNA differences as?
Go ahead.
So the big difference then between my view and then the evolutionist view, for example, is that we explain the vast majority of nuclear DNA differences, not by mutation like we explain the mitochondrial DNA, but as pre-existing or created heterozygosity. So that one genetic difference, Aaron, has massive implications for, one, the timing as to the origin of species, the function of the genome, okay? I would agree that if you're just trying to explain the origin of the species, or you're looking at coalescence calculations in mitochondria and nuclear DNA, just by mutations, then we're going to need millions and millions of years for all these differences to accumulate. I totally agree with you on that one. But if the differences are there from the start, I'm almost done here. The simple operation of recombination or even gene conversion and some mutation can produce visible variety in even a single generation. So that actually correlates perfectly with the coalescence time of mitochondrial DNA and the coalescence time of nuclear DNA.
So, trying to understand your position here. Are you trying to say like a heterozygosity, like some sort of perfect heterozygosity, where you have every single possible variant that could be expressed within the genome of Adam and Eve?
So by pre-existing functional heterozygosity, I would say that the DNA differences, the millions of DNA differences that we do see across species, I mean, we have DNA differences within our own genome, and then DNA differences between, like, for example, me and my daughter would have DNA differences in us. So I would say that the vast majority of those DNA differences were created as functional DNA differences. So yeah, they would be, there would have been tens of millions of heterozygous DNA sites at creation. That's why recombination and gene conversion, right, kind of like shuffling a deck of cards, could reveal genetic variants in just a single generation. But that's kind of a counter to your argument and statement that the nuclear DNA has a coalescence calculation that goes further back than the mitochondrial DNA. Go ahead, take as much time as you need there. I know I talked a lot.
So for one, I don't see how that would be different than the cryptic variation, which I was describing that we know occurs. Also, I'm still not too clear. Like when we talk about heterozygosity, we're talking about allelic variations because we're diploid organisms. We have two versions of genes typically. Just trying to kind of hone in, be more precise about what you're trying to get at here. Let's talk about, say, hair color as a trait. For this perfectly heterozygous couple, are you saying that between the two of them, they would have alleles for every single variation and type of hair and hair color known to man in their genome?
Good question. So a lot of the variants that we see, for example, like blue eyes, for example, that, because we can look at allele frequencies, right? So the more rare an allele is, the more rare an allele frequency is, we can conclude that that was the result of mutation. Okay, so we would say that that was the result of mutation probably after Babel. Once there was the dispersion and a lot of people groups became isolated. But yes, the majority of them that I would consider design variants or just built-in alternatives that God would have put into Adam and Eve. I'm saying that all these built-in alternatives can be scrambled over time and therefore reveal new traits and new species quickly.
So for eye color, I'm just not sure why you would need that, because we know there's two different types of genes currently that produce a continuum of eye color across populations, and it's also related to hair color. So I don't see why you would need all this variation of. It seems kind of too simplistic. I don't know. I'm not following what you're. It doesn't seem to make sense with genetics.
Well, I'm saying with built-in alternatives, with this functional heterozygosity, I mean, we can see changes through, I don't know, we can see changes through homologous crossover, changes with jumping genes, right? These retrotransposons, the ALUs, for example, other mutation-related means as well. Recombination can generate diversity quickly. I mean, we can see the echo of this though in breeds.
You don't need perfect heterozygosity for novel variation to occur. Like it's just not necessary. There's lots of other mechanisms to provide variation and novelty in phenotypes without having to have some huge swath of like some gigantic genome full of hundreds of thousands of genes, way bigger. I mean, I don't even know how Adam and Eve's genomes could have held all these variations with it wouldn't even been a human genome compared to what we have now. It would have been something totally different. So
Well,
it's just not necessary. Like, look at the structure of, we know for a fact through experiments, uh, say you can take the hold on, let me try to think of the best way to explain this. Very minimal genetic diversity is required for large phenotypic diversity. And that's because of things that occur um, that aren't from protein coding genes, like regulatory
right.
genes. It involve timing and place when something happens, how it's expressed, how much is expressed. You can take the limb bud from a chicken that's going to form like a bone or like a leg and transplant it to its head during development, and it'll grow a fully functional leg on its head. Um, so it doesn't really take a bunch of genetic variation to create like novel traits. You look at the morphology of a bat wing versus a bird wing versus a pterodactyl wing. What do they all have in common? They all have the same bones. They have same wrist bones and appendages. The bones are just in different lengths. Like you got the one for the pterodactyl, you've got, I think it's this thumb digit that goes way down here. So in development, bone is like a leading event that occurs, and then from that leading event, you have what's called exploratory behavior of muscle cells and nerve cells that will kind of all kind of shoot out in this exploratory behavior. So it's not guided; they just kind of go out in different directions. And once a nerve cell makes connection with the bone, then the other ones disappear, and that's the connection that's made. It's kind of like a fly flying around the house, lands on some fly paper, gets stuck there, and then that's it. There's no like, it was just randomly flying around and it gets stuck there. Or like ants when they come out of their nest, they have exploratory behavior. They're looking for food. They all go on various different paths looking for food. It's not like they have some plan or something. One happens to find like a dead cockroach, right? And now it comes back after eating the cockroach. It leaves a trail that the other ants can follow. Then they all kind of converge on that one trail, and that makes that one connection there, right? So in development, there's lots of different pathways where you can have lots of diversity form, like in different types of structures,
Right.
like the wings and bat birds and pterodactyls that don't require a bunch of different coating protein genes and heterozygosity to occur.
Are you saying that there's a lot of function as well, and there's a lot of correlation between the non-coding DNA and the actual protein coding DNA, right? The regulatory elements when it comes to the function, when it comes to the changes within our genome. Is that pretty much, in a nutshell, what you're saying?
Yeah, a huge part of the genome is within like these regulatory regions
Right, I
that
agree.
have effects on these coding regions that code for proteins, for example. So they play a major part in developing the phenotypic diversity, especially like I said during development.
Right.
You can just take for example, there was an experiment done. I can't remember who did it. It was where they took like the germ layer that forms the bottom side, like the belly of a eukaryote, and transplanted it to the back to see what would happen. And then actually it formed a completely separate embryo, like fully, differentiated into all the different cells, because all that information was there. It just depended upon the timing of those regulatory regions to kick in and make this occur. So very, very minimal variation is needed on the genotypic level to create lots of different novel phenotypic variation. You don't need created heterozygosity.
Well, I actually agree with everything you're saying in regards to those types of changes, right? The hidden information that can reveal these types of traits. And a major prediction based on the created heterozygosity model. For one, everything that you were describing, I believe, is consistent and actually helps our model out even more, actually. And when it comes to the created heterozygosity, I think to make it a little more simple and to keep it genetically speaking, say for the audience, I mean, as you were saying earlier with the alleles, right? It goes back to this idea of, you got the big A, big B, little b. So what I'm saying is if Adam and Eve and the created kinds were created genetically, let's say they were created genetically homogeneous, right? With absolutely no variety in their DNA, say all capital letters or all lowercase letters, then yes, I'm saying it's going to be difficult to create variety in just a few thousand years. So according to our model, we do look to created heterozygosity to explain the rapid speciation, for example, right? So let's say, let's use the kinds that God created, okay? Let's say he created them with capital A, lowercase a, capital B, lowercase b, and then you do that, Aaron, for the millions of DNA positions that actually exist in our genome. That means they have the potential to produce all sorts of variety in just a single generation. And then everything you said actually helps that hypothesis, because now we know the non-coding DNA, as you were talking about, it functions much like a computer. It's involved in regulatory functions, right? For the audience sake, that may not be as science minded. That's just when and how fast to make specific proteins, for example. Embryonic development, the timings of protein manufacture. I mean, there's so much that's going on in the coding and non-coding regions, and even DNA elements, for example, such as the endogenous retroviruses, such as the ALUs, the retrotransposons. I mean, we're seeing more and more function, more and more ways to generate change in the genome that's all consistent with this hypothesis that I'm talking about. And obviously we've kind of gone down a bit of a rabbit trail, because I was just trying to explain why the coalescence would be different in nuclear DNA versus mitochondrial DNA. I know I talked a lot there. Sorry, I like what you're saying, so I wanted to kind of piece it together, the bigger picture, right?
Well, real quick with what you're saying, and we can move on to chromosome number 2 if you want to, but like with what you're saying, with this created allelic variation it contains all the different the landscape of potential phenotypic variation. You don't leave any room for these regulatory regions to exist. You totally squeeze them out. So now you're gonna have to expand the genome of every single animal to these just vast proportions. And that's just based on a misunderstanding of how variation occurs and how novelty develops and how new phenotypes develop. So I can see where you're coming up with your idea, but I think it's just based on not a very knowledgeable understanding of how it all works.
Well, I mean, how much of the genome, both non-coding and protein coding, would you say is functional on, say, a percentage level?
I don't know, but I don't think it's relevant to what I'm saying.
Well, no, I believe it is, because from my understanding and from the literature that I've looked into and read, the evolutionary community, and I've actually debated many different scientists, Dr. Stefan Frello, for example, who's well known in this controversy, they will say the genome can't be any more than, and we're talking about regulatory elements as well, like you're talking about, any more than 15%, maybe max 20% functional. But we're saying based on this created heterozygosity model, nearly the entire genome is functional. Therefore, what you said about fitting all of that in the genome, for example, I think that's based on an evolutionary assumption that the majority of the genome is based on evolutionary leftovers, genomic fossils, right? The pseudogenes, for example, the endogenous retroviruses. I mean, it was Richard Dawkins who said that 95% of the genome may as well not even be there. All the parasites and things that are infested in the genome. But we now know that is just completely false. I mean, paper after paper. I mean, are you aware of even these endogenous retroviruses that are shared among like humans and primates and is used as evidence for universal common ancestry? Are you aware of the accumulating evidence suggesting that they're incredibly important in the regulation of genes and determining cell types? I mean, just more and more function every day. It's amazing, actually. I love it.
Yeah, that's no surprise. You expect co-option to occur. I mean, this is the way DNA works. If you have, like I was mentioning earlier, the read through stop codons they'll produce different variants of proteins that have different terminal ends, and some of those may be lethal, and so they're non-starters. They get selected out, but you have enough different variants that as soon as the selective pressures become enough, and you get enough of these buildup, this capacitance of these swaths of structural variants, then they can be selected for. Hold on, I lost my train of thought. What was the last thing you said? Sorry.
Well, just about the endogenous retrovirus, I do want to say, because I
Yeah.
noted that you said that they were co-opted, but to be honest with you, I have to say that that's just nothing more than mere uninformative gloss, and I'll tell you why. Because the evidence shows that these endogenous retroviruses are incredibly functional, right? They have crucial functions that help regulate genes. They even help to determine cell type. So the evolutionists, and I read through these
Yeah,
papers,
it's not a problem. It's a universal code.
well, no,
It's not a problem.
but the thing is, what I'm saying is, far from being junk DNA, as evolutionists have always assumed, right? Richard Dawkins, for example, have always assumed that 95% of the genome may as well not even be there. Now we know that the pervasive retrotransposons, the ERVs, for example, that populate our genome, they have a powerful capacity to influence genes and chromatin. So now the evolutionists, I'll read through these papers, and they go through all these various functions that are being discovered, and they always end it with, okay, these functions were co-opted, but these functions were not observed to be co-opted. And I challenge you to provide a paper afterwards that show an endogenous retrovirus going from non-functional to suddenly incredibly functional in embryological development. It's just not there. That's the evolutionist rescue device to explain away the evidence in the functionality of the genome. So I mean, if you have some papers that show these DNA elements, the co-option actually occurring empirically, then you may have an argument there. But I've never actually seen that demonstrated in these papers. Do you have any evidence suggesting they're actually co-opted, other than just saying they're co-opted?
No, I'd have to. I'm not going to lie and say, yeah, I got this certain paper in mind that I'm going to produce, but it's not. It's not. I don't see any problem with it. I mean, you could have an ERV occur in a regulatory region that would affect, say, the production of like BMP, bone morphological protein, which would like inhibit certain developmental pathways, which could be lethal, but it could also create variation that could lead to some novelty as well,
Right.
and therefore that sequence that was where this ERV was kind of spliced in could then have some sort of new novel function within the genome. I mean, it's all the same base pairs, it's universal code, so I don't see any problem with these things being co-opted. Like there's no contradiction there. There's nothing logically impossible about it.
Right. I'm just saying that it's my position that because I read the literature, right? I read these papers on the functions of these, say, transposable elements, for example. I mean, they really are a incredible and important source. They're tightly regulated, they're biochemically active, and they are non-coding elements. I mean, this is a direct prediction based on the pre-existing heterozygosity hypothesis, suggesting that the vast majority of our DNA differences, the vast majority of our DNA elements are incredibly functional. I mean, they function in the transcription, in transcription factor binding sites and in non-coding RNAs. And I understand that you may have because I noticed a few words you said, like they could have, or maybe this, or maybe that. There's just, and I challenge even anybody listening in the audience, provide me a paper that shows a non-functional transposable element or a non-functional endogenous retrovirus suddenly going from the non-function to something that's incredibly important in, embryological development or immune response
well, I think you're also not realizing when you're talking about function, you kind of seem to have this black and white, like function, non-function, doing something important, not doing something important. but it's much more complicated in that genes have variations in the amount of their expression. So you could have a transposable element that does nothing at first, but then it can accumulate some mutations over time. And like I said, within a certain environment, it's cryptic variation that can be then selected for, and it can have some very minimal level of expression that has some sort of function relative to the environment.
Right.
It's not just some all function or no function. It's different degrees of function.
Well, and I understand that that's the evolutionary story and that's what you believe, but
It's
I
not a
definitely don't want
story.
to believe.
This is
Well,
empirically
it is.
verified through
It is a story
research.
because these very important functional roles in these DNA elements, the evolutionists just said they were co-opted. That's nothing more than storytelling. I'm sorry.
Just get onto PubMed and look up papers that show variations in expression of genes and regulatory elements. You'll see
Right.
a bunch of different papers written on it. It's not just so storytelling. I mean, this is empirical evidence that we do in the laboratory.
Right. No, and I understand your position. And I appreciate your response. I think because we have been going over for over an hour, let's, if you want to, we can, I think, because the only thing really that we haven't touched on that was talked about to make for a complete discussion would be the human chromosome 2 fusion. We could touch on that. And
I
then maybe,
actually wrote some notes on that because I thought I was going to have like an opening statement. So I did jot some notes down before we were going to get together.
And I think that would probably be fun for the audience, fun for Tom. That's what it's all about, right? Having a good. I've had a lot of fun. Maybe this will be a good way to end it. We'd probably end it around the hour and a half mark, and maybe even set up another discussion because there's just you're right. There's just so many different things to.
Can you guys give me like 2 minutes?
Go for it.
Come right back, and then we'll just kind of wrap it up. I'll go over my notes on the chromosome 2, if that's okay.
Yeah,
beautiful.
For sure.
I think that's a perfect way to end it. So, Tom, how we doing?
Good. Doing good.
You got any big debates planned soon?
I think so. I think I just set up like 6 of them yesterday, and I don't remember what they were anymore.
How many debates have you done? Do you keep track, or?
I think I'm at like 70 videos on YouTube or something. I don't know.
Nice, good for you. Have you tried to reach out to like Answers in Genesis? I'd love to see you discuss, testable predictions with Nathaniel Jensen. I know they're difficult to get a hold of and to schedule with, but have you reached out to them at all?
Yes, I think I reached out to them twice. I think one time I didn't hear back, and one time they said they were too busy. But they have a really complicated system that you have to use to get to actually email them. You have to specifically say, I want this speaker at this date, instead of just like, you just do a general inquiry as, are any of your speakers available to talk about blah, blah, blah. So they make it really complicated to try and go through the system to actually to get their speakers on board, which is difficult.
Yeah, it sucks because there's definitely a lot of scientists on staff there that I'd like to, see you discuss with. I think that's what's so good about Ken Hovind is he runs an independent ministry, so he's so easy to schedule debates and interviews and discussions. He's literally just a phone call away.
Yeah, I definitely like to talk with Behe and Myers and Dembski. Those guys I definitely want to talk to.
Actually, next Thursday we're having Paul Nelson. Is his name. He works for, I believe it's Discovery Institute, or he works alongside Stephen Mayer, for example. So I'm going to interview him next Thursday, but if you could actually ask him if he'd be willing to come on my show, that'd be great. I will. Yeah. I think that would be an interesting discussion as well. All right. Actually, I'm just going to grab a bottle of water real quick. I see that Aaron's back. Just 1 second.
So Aaron, would you want to give your brief introduction thing and then just go from there?
Yeah, you don't mean to wait for him, or I guess he can.
We should probably wait for him so he knows what we're talking about.
Yeah.
Oh yeah, I'm here. No worries. If we want to.
Okay. And so I just jotted down some notes. I hope it's not too long. I'll try and read through it really quickly. Hopefully it's all coherent. Just for my examination of human chromosome number 2, I think it's probably some of the strongest evidence we have for common ancestry, at least with humans and the great apes. So.
Right.
All right, so here we go. So as we know, like humans have 48 chromosomes, or humans have 46, the great apes have 48, and we know there couldn't have been a deletion event because that would be lethal. So there had to have been some sort of head-to-head telomeric fusion of 2 chromosomes in our Ancestry that provide us with our 46. Kim Miller did a good job of going over this, and his Dover versus Kitzmiller trial, which kind of left creationists ever since then scrambling to try and find ways to deny this evidence, which is kind of funny to me. But so just some of the good evidence from it, the way that we know it's actually a fusion event is so it has two centromeres whose surrounding sequences match up perfectly with the sequences of that in the ape pair, which is 12 and 13 in the great apes. The end of the telomeres, a repetitive sequence of TAAGGG, is butted up against CCC-TAA, which is to be expected from an inversion of the other telomere, which is predicted. The synteny and order of genes around this fusion site has been largely preserved. You can just go on to the ensemble.org, like I mentioned earlier, and look at it for yourself. You can look into this database and you can see that order. And you can actually see the genes surrounding the fusion site. I won't go over all of them, but on the left side, you've got a gene that's coded CBWD2, and then you got the fusion, and then to the right, the other strand, you've got gene RABL2A, and a whole list of genes that all match up on the opposite sides of this fusion site. And the genes to the left of the fusion site are found on one end of the chimp chromosome 2A, while those on the right they match 2B perfectly. Hold on a second here. So the chromosome 2 story is simple, it's clear, it's understandable, it's powerful, which is I think why creationists have scrambled to find ways to deny it. You'll hear criticisms of it. They'll say, like in Sandy, you may present these. The telomere site is too short. Like a typical telomere has over 1,000 repeats, while the fusion site has less than 200. But that's to be expected, since one of the functions of the telomere is to prevent chromosome fusion. We know that telomeric disorders occur and reduce repeats. When this happens, fusion becomes much more common. You'll hear repeats are degenerate and don't perfectly match the TTA-GGG pattern. Some are 5 bases long, some are 7 bases long, but that too makes perfect sense. Telomerase is the enzyme responsible for producing the 6 base repeats, and that only works on the tips of chromosomes. Once they fuse, they are subject to changes that degrade and sorry, I can't read my own handwriting. They degrade the repeats. The mutation and recombination changes the number of repeats. Since the fusion occurred several million years ago, a degeneration pattern is exactly what we would expect to find today. Another claim put forth by the Institution for Curation and Research is that an active gene, the DDX11L2 gene, spans the fusion site and therefore falsifies the fusion story. It doesn't. Go on to Ensemble, pull up chromosome number two, zoom in on it, look at it. It does not span the site. And this claim ultimately backfires anyways, in a most revealing way, because it's largely been abandoned now. The DDX11L2 is part of a family of pseudogenes, 18 in total, scattered around the human genome. Now guess where 17 of these 18 are located? Right next to a telomere on other chromosomes. The exception is chromosome number 2. Okay, and this is profoundly compelling evidence that it too was once part of a telomere, just as the fusion story demands. It gets even better though. Next to each and every DDX11L pseudogene is another gene designated WASH, W-A-S-H, located on the side away from the telomere. There's also one in the middle of chromosome number 2, next to the DDX11L and the fusion site, as to be expected. Finally, in every 18 locations on the human genome, the DDX11L pseudogene is transcribed in a direction away from the telomere, while the WASH gene is transcribed in a direction toward the telomere. The same is true for the copies of the genes near chromosome 2, the fusion site. In fact, every landmark surrounding DDX11L2 gene suggests that it was once located next to a telomere. And so this doesn't represent common creation, because all these different sequences I mentioned represent events in time that can only occur through common ancestry. I'm done.
Okay, so I appreciate that. I guess I do want to try and address everything you just said, so I hope I can have equal time here to do so. Okay, so just to be on the same page, Aaron is claiming that the human chromosome number 2 then arose by the fusion of 2 chromosomes in the hypothetical chimp-human common ancestor. So right off the bat, you would have had the split roughly 6 million years ago from the common ancestor of chimps and humans, and this so-called chromosomal 2 fusion would have occurred in the human line. Therefore, it would be indirect evidence anyways, as a chromosomal 2 fusion could have easily been passed on, say immediately post-flood or post-Babel, and actually, since we believe in a population bottleneck, could have been passed on quite easily. So I don't see it as very good evidence for universal common ancestry, since it can be explained in either model. So that being said, I will go on to the fact that, fusions, they do happen in nature. So that's a fact. And therefore, the resulting fused chromosome can then be transmitted to the next generation. But it's also a fact that those fusions, those types of fusions that occur in the germline, they are very deleterious, right? They're going to reduce fertility. They're going to disrupt chromosome architecture, for example. Therefore, selection should be selecting against those types of events. I would say fusions in general are examples or manifestations of genetic entropy consistent with the fall, consistent with biblical creation. But here's the thing to consider. Most germline mutations, right, fusions that would have to be passed on, which is exactly what this type of fusion would have to be, they don't actually arise as end-to-end fusions, but instead they arise due to it's mistakes in the crossing over process. So I've got a lot of papers showing that chromosome fusions do arise within cancer cells end-to-end, which is what this, from what you said, it's an end-to-end fusion. But those that occur in cancer cells, Aaron, those are not transmitted to the next generation, obviously. Those are in the somatic cells. It has to be in a germline cell. So crossover fusions, they then result in the deletion of the ends of each fusing chromosome. So that means that the special telomere sequences at the ends of both chromosomes would be deleted, and any genes in the crossover region would also be deleted. So that being said, there wouldn't even be any trace of a sequence such as the reputed fusion site, right? Because no associated subtelomeric sequences, and then there would be no trace of internal telomeric sequences. But here's the problem, is that the so-called telomere to telomere fusion, it would have to occur in the germline, okay? Mm-hmm. When it was first discovered in 1991, it wasn't exactly what they expected. It had a different pattern, for example, than what was found primarily in living mammals, that it actually went through a chromosomal fusion, okay? Now, it didn't contain something called satellite DNA. So I noticed that wasn't something that you mentioned, because yes, the telomeres are too degenerate, but certainly that can be explained based on the shortening of it, because the telomeres, as you said, the telomeres are the end caps of chromosomes. They prevent fusion, right?
It does have the satellite DNA, by the way.
So is your position that the human chromosome 2 contains satellite DNA?
Yes, that's what's in the literature, and just pull up Ensemble, look at the human genome, and see it for yourself.
Well, because from my understanding, they find satellite DNA combined with satellite DNA in observed cases, or satellite DNA combined with telomeric DNA, but telomere to telomere fusions, the one that's being purported in the human chromosome 2, is rarely if ever, heard of or observed in nature. I'm not talking about like cancer cells or, so it is your position that the human chromosome 2 fusion, the telomere to telomere fusion, satellite DNA was discovered? Because I haven't seen a paper that says that.
Yeah, it was.
Well,
And
I have,
we know this has occurred. It happens commonly in human chromosome 13 and 14. There's family in China, lineage in China that has this
right.
fusion. People live and they survive and they produce children who have this fusion of head-to-head fusion, telomere fusion of chromosome 13 and 14. So it does occur.
Well, I'd have to see the actual paper demonstrating that satellite DNA was discovered in the human chromosome 2 fusion. I am going to say that it wasn't. But even those internal telomeric sequences that you were talking about, right? Because the telomere regions, the remnant telomeres that should be found, they are highly degenerate. Okay, they're a lot smaller than should be because
Yeah, and
telomere sequences. Yeah, no,
what should be expected.
but yes, I understand that you're saying that should be expected, even though telomere sequences are what, between 10,000 and 15,000 sequences long, like base pairs long. Because you're saying because it happened 6 million years ago.
No, over 1,000 or so.
Okay,
and,
so
at the fusion site, the telomeric region repeats are about less than 200.
Right, and I'm saying that those internal telomeric sequences that are found, like I think it's 800 bases in length, for example, or even 700, and I think it's only like 70% similar to what would be
it's less.
fusion.
It's less than 200.
Right. Okay. So you do agree that it's highly degraded, but you're saying that this should be expected technically.
Right. Yeah, that should definitely be expected. If it wasn't that way, then that'd be weird. We'd have a problem.
Here's the thing though, those internal telomeric sequences that they actually look to and say, okay, here's the remnant telomeres that are highly degraded, they're actually found all over the genome. And there's many papers, and I can send them or post them afterwards, that have been published that have given us functions for many of these sites. For example, they're binding to things called, as you know, transcription factors, which then initiate the copying process of a gene into RNA. So that's not just unique to the human chromosome 2 site. So the fact that it's degraded and then they've tried to explain it away,
you're wrong. The TTAGG sequences, repetitive sequences, are only found on the tips of chromosomes, and the wash genes are only found right next to those that I mentioned earlier. The DDX11L2 does not span the fusion site.
Well, real quick, before you go on to that, what I'm saying is those internal, are you disagreeing with the fact that those internal telomeric sequences that you're pointing to that are seen as highly degraded in the human chromosome 2, are you saying that they're not found all over the genome with important functions?
No, the telomeric repeats are only found at the tips of chromosomes. That's the only spot they're found. And that's the same place the DDX11 genes are found only at the tips of chromosomes. The only anomaly is in chromosome number 2, right there in the center where it shouldn't be. And what's next to it? Wash gene, where it also shouldn't be. What else is there?
But you're only looking, but with these so-called remnant telomeres, you're only looking at something that is, well, you're saying 200. I thought that it was, okay, so the fusion site is 798 bases, but you're saying the remnant telomeric sequences are about 200, right?
About 100, 150.
Well, for one, I mean, the reputed fusion site being only 798 bases seems to be an extremely tiny signature for such an event anyways. But I'm saying that those 200 sequences, those sequences that you're looking at, I'll probably just have to provide some data after the fact. Maybe you haven't heard of it. But those internal telomeric sequences you are pointing to as evidence for remnant telomeres are actually found all over the genome with many important functions. For example, the transcription factor relating functions. So that is not, and you mentioned I think it was the centromere, the cryptic centromere site. But that, which you should find, obviously, one would have to be deactivated if there was a chromosomal 2 fusion area. That would be lethal, of course. So that should be found, I agree.
Yeah, one's dominant over the other. That's what typically occurs in fusions.
How many, so this cryptic centromere, how many bases?
It's not cryptic. It's just a inactive.
Deactivated. Okay, so a deactivated. How many bases is it that they've pointed to as the remnant centromere or deactivated centromere?
I'd have to go onto the database and look. I don't know.
I think, from my understanding, I don't have notes on it here, but I believe it's less than 200 as well. So that itself is also very small. But that, and I understand you were talking about the reputed fusion site being spanned by a functional gene, but I'm not even going to point to that right now. The cryptic centromere, or the deactivated centromere, as you'd want to call it, it's way too small, for example, that region. It's too human to actually be a remnant centromere, and this entire
human lineage.
Well, just 1 second. Here's the big thing. That entire sequence is internal to a functional gene, and I'm talking about the so-called deactivated centromere. So that would be impossible if the deactivated centromere sequence was ever a real centromere, as it would have had to be if it was a chromosomal 2 fusion. So, I mean, how do you explain that fact?
I've never heard that one. I'd have to look it up. I don't know. Never heard about a gene spanning the centromere. That's what you're saying?
Yes. Yeah. I'll send you the I'll send you the paper on that. I want to say
I would assume it's like the claim that the DDX11L2 spanned the fusion site when it didn't. I mean, I know that there's millions of dollars involved in trying to figure out ways to deny this, and paying these PhDs who don't have relevant degrees to try and find a way to discredit this. But statistically, too much adds up. Like, how do you even explain the simplicity of moving from where the fusion site, moving from TAA-GGG, but it directly up against CCC-TAA? How does that occur?
Well, before I go on to that one, because you were saying that it doesn't span the fusion, are you using Ken Miller's argument that it doesn't span the fusion site?
I'm using my own eyes to go onto ensemble.org, pull up human chromosome number 2, and zoom in and look at it for yourself.
Right. So that exact argument, and just because, for lack of time and also for lack of just going on and on, Ken Miller used that exact same argument, and him and Dr. Tompkins, Jeffrey Tompkins, they had a back and forth through email. And Miller, he attempted to explain the longer transcripts of the DDX11L2 gene. In the paper, what does he say? He says that it's easily explainable by variability in the termination of transcription, so that occasionally a somewhat longer RNA is produced. And he says that, let me see here. He goes on about how you're saying that it's not actually spanning the fusion site, like the functional gene, for example. And Dr. Tompkins goes over why that's wrong in this paper, that I, instead of just reading off, I would probably just send it after the fact and kind of see what because Dr. Tompkins talks about how the longer transcripts begin ahead of the fusion site at the 5' end of the gene, and that it's well documented fact that transcription begins at the 5' ends of genes. So he's pretty much just going into the fact that it really does, this functional gene does span the fusion site. But I just have to urge you to maybe read that paper and see it's a newer paper, so I don't know, maybe you haven't seen it yet.
I apologize, just to interject for a second. The people in the chat are saying that, Standing, you're getting a little loud. Could you talk a little bit quieter to try and balance out the volume a little bit?
Oh, yeah, sorry.
Thanks.
Am I just getting loud now?
Yeah, you've been getting louder for the past 3 or 4 minutes. I think it's just you're getting more energetic or something. So it's not.
That makes what he says more true.
Yeah,
the louder and the okay. Well, I will say this: it is getting kind of late. It's been a lot of fun. We can do a couple more of these. Did you want to kind of sum up your position on that there, Aaron, and then we can kind of go from there?
Yeah, I mean, it's, to me, it's the cumulative case. Like, there should be no reason for all the other features I mentioned to perfectly match up from chimp chromosome, ape chromosome 13 and 14. If you flip them around, there should be no reason why we should see the exact same sequences meet up in the middle of human chromosome number 2 with the exact same genes flanking the fusion site. If common ancestry makes perfect sense of this, creationism, it's an utter mystery. It's just a complete mystery, and you have to just scramble to conflate some sort of
Well,
stories to
sorry to cut you
describe.
off because Aaron, you were saying that the sequences that flank the reputed fusion site, they should match up with sequences near the ends of the corresponding chimp chromosomes. And you're
Yeah,
saying
and like I said, even the telomere sequences, the CCTAA that
right.
flanks, that comes off of butts up directly to the TAA-GGG from the left side. There's no other explanation for that, I mean, other than
But
it's a fusion, and unless you want to say that's just the, I mean, you can always appeal to God, say that's just the way the designer made it, and he's trying to be deceptive if you want to, but just to make it to fool us for some reason. But I just don't see any reason for that to be the case.
It's my understanding that the ends of the two corresponding chimp chromosomes that we're talking about, they haven't yet been fully sequenced. So if they haven't been fully sequenced, and then the actual region that contains the reputed fusion site now
They have
DNA.
so on the left
Real
side.
quick, because that region that contains the reputed fusion site that we're talking about, that you're saying is derived from two ape chromosomes, now appears to be DNA that is uniquely human and then is absent in all ape genomes. So there's not this perfect synteny as you were.
No, there is. I actually went on Ensemble earlier. I wrote down 4 genes to the left, 4 genes to the right of the fusion site. You've got ILIRN, and then you've got PSD4, and then you've got PAX8, and then you've got CBWD2. Then you've got the fusion. Then you've got RABL2A. Then you have SLC35F5. Then ACTR3, and then DPP10. And these all match up perfectly with the chimp chromosomes near the telomeres on both of the separate chromosomes.
Yeah, if you could send me, because I think in regards to the chromosomal 2 fusion, we're definitely going to have to send each other some of the things we're discussing. I'll send you the paper I was talking about, but it's my understanding that entire region of human chromosome 2 doesn't actually appear to have homology with any part of the 2 chimp chromosomes that are actually being said to have fused. But that region, and it is a very sizable region within the human chromosome 2, only aligns with various other chromosomes of the chimp genome. I think maybe like chromosome 9, for example. So there should be more so perfect homology and synteny. And I understand that you're disagreeing with what I'm saying in regards to the synteny, but that's my understanding. And we may just have to exchange some papers after the fact. But I do want to say, I believe that there are some discrepancies based on satellite DNA, degenerate telomeres, the so-called deactivated centromere. But that being said, I'm also open-minded to there being an actual chromosomal 2 fusion, because that's why I started off my section on this, I guess, by saying that fusions in general are manifestations of genetic entropy, and we could easily explain a chromosomal 2 fusion in the human line post-flood, post-Babel, especially because we believe in a bottleneck. So I think either way, if there was a fusion, it's indirect evidence, and therefore it's agnostic to the debate. That's just my position on it.
Yeah, it still doesn't explain why we have the same exact ones in the ape chromosomes that match up perfectly. That shows history. That shows a history of events occurring in common ancestry. That's the only way it can be explained.
Well, I'm saying that the synteny is not lining up perfectly. Plus, we also know that synteny, right, like the gene order and the architecture of the chromosome, are also playing in very important functional roles across different species. So there's so many multiple overlapping codes in our genetic information system. There's so much hidden information, compressed information. So, it's my position that we understand very little of the DNA language, and a lot of the best arguments for universal common ancestry, whether it's the human chromosome 2 fusion, pseudogenes, endogenous retroviruses, for example, or DNA level nested hierarchies, I believe that that's all being thrown out with the junk DNA paradigm, because more and more data suggests that our genome is incredibly functional, and I believe that that's all consistent with our model of created heterozygosity. So I definitely don't want to beat a dead horse there.
Yeah.
I appreciate you doing this discussion. I'd like to give you the last words on it, and then maybe in the future we can set up another discussion, because it's been a lot of fun, Aaron.